Ladies and gentlemen, will the congregation please rise?
Good evening, and please be seated. I want to say relax, but there is an inaugurating professor here. So. Good evening, everybody. It's wonderful to see all of you in the Senate room, and also welcome to those who are joining us online. My name is Inaugural. I have the privilege of being a Deputy Vice-Chancellor, and in fact, I'm standing in for the Senior DVC, Professor Ruxana Osman, this evening. So forgive me if I'm not quite as charming as she would have been. My job is very simple. It's to introduce to you the procession. And in the procession, we have the Dean of the Faculty of Science, Professor Nithya Chetty. We have the head of the Evolutionary Studies Institute, Professor Marion Bamford. We have Professor Jonah Chouanier, who we will hear from later giving the vote of thanks. And of course, we have the star of the evening, our inaugurating professor, Professor Jennifer Botha. Folk, an inaugural lecture is a real rite of passage as an academic. And it's that moment when a professor stands up, and no pressure, but when a professor stands up and professes. But I think equally importantly is for that professor to just allow her loved ones, her family, and her friends to know what on earth it is that professors do on a day-to-day basis to drive knowledge and to really grow institutions like this one. So thank you all very much for being here. It's now my pleasure to call on the Dean of the Faculty of Science, Professor Nithya Chetty, to introduce our inaugurating professor, Professor Chetty.
Thank you very much indeed, Professor Inaugural, Mr. Deputy Vice-Chancellor, also the Director of the Evolutionary Studies Institute, Professor Marion Bamford, colleagues and friends. So delighted to be here this wonderful, almost spring evening. It feels that way after the cold temperatures. It is my pleasure then to present Professor Jennifer Botha, Director of Genus and Professor in the Evolutionary Studies Institute in the Faculty of Science, to deliver her inaugural lecture on the topic of tracing impacts of mass extinctions. Now, understanding mass extinctions and predicting the future ones requires analyzing affected organisms. The Triassic period, bookended by two major extinction events, saw the rise of mammalia forms and dinosaurs, making it ideal for studying survival strategies, clade origins, extinction patterns, and ecosystem recovery. South Africa offers exceptional fossil records from both the Permo-Triassic and end-Triassic extinctions. Here, Professor Botha will compare the life of history trace of species that survived these events to explore patterns of resilience and evolutionary innovation. Professor Jennifer Botha earned a PhD from the University of Cape Town, we'll forgive her for that, in 2002. Following a postdoctoral fellowship at the Zico Museum, she became head of the Karoo Paleontology at the National Museum in Bloemfontein. In 2003, she joined the Evolutionary Studies Institute here at the University of the Witwatersrand and was appointed Director of Genus, the DST NRF Center of Excellence in the Paleosciences. Her research focuses on paleobiology and paleoecology, particularly how extinct vertebrates responded to environmental changes. By studying fossil bone microstructure, she investigates species survival during past mass extinctions, which are a critical context for today's ongoing biodiversity crisis. Using past mass extinctions as models, she used a variety of techniques, including biostratigraphy, morphology, and osteohistology, to test theories regarding differential species survival during mass extinctions. This work is especially relevant as Earth faces a possible sixth mass extinction driven by human activity. Paleoscience plays a vital role in understanding these patterns, revealing that only by studying the past can we fully grasp the present and make informed decisions for the future. Mr. Deputy Vice-Chancellor, I present to you Professor Jennifer Botha for her inaugural address.
Thank you, Nathia, and thank you everybody for being here on this special occasion this evening. I'm going to try and do this without my glasses. Right. You may think with a picture like this, you've come to the wrong talk. But I want to go back to when I was a teenager in high school. I actually wanted to be a marine biologist. So that's why I went to UCT, it's by the sea. And I'm sure I'm not alone. When we are young, we think of these idealistic pictures of playing with dolphins and rainbows and everything being sunny and beaches. And so that's why I went to the University of Cape Town to become a marine biologist. However, much to my shock and horror, I found out this is pretty much what most marine biologists do. A lot of statistics, a lot of maths, a lot of seeing viable fish populations for fishing companies, and I decided I actually didn't like fish that much to spend the rest of my life with them. So I said thanks, but no thank you. I wanted to do something else. And it was during my honours year that my supervisor, Professor Anusia Chinsami-Turan, joined our department at UCT. Before then, we hadn't had any paleontologists in our department, so we were obviously taught evolution, but there was nobody really passionate about fossils or opening an entire new world for us, and that's what she did for us. And I was completely amazed. It was something so new to me, and it opened up a whole new world, and I decided, well, if I can study fossils, paleontology for the rest of my life, I will be happy. So that's what I did. I joined Anusia for a master's, and then I went on to do a PhD at UCT with a nice floppy hat. So you can see it's not a Wits do. And this was in 2002. After that, I went and joined Professor Roger Smith at the Ezeco Museum in Cape Town. And although my postdoc was only 2 years, because I had spent quite a while in Cape Town, I probably spent about a decade with Roger Smith going in the field, learning how to do field excursions, and he's taught me so much that I know about field excursions and field trips. And this is Roger over here. You don't have to actually get this messy, by the way. We had a plaster fight, but at least we know how to have fun in the field as well. But it was time to move on, and I was offered a position at the National Museum in Bloemfontein. Who was then running the museum was Rick Nuttall. He was the director of the museum. And I just want to especially thank him today, because if it wasn't for his support and his constant motivation and positiveness, I wouldn't be where I am today. He was a fantastic director and manager, and he really did give me a lot of support. But it wasn't just a short stepping stone. I stayed there for 18 years, until more recently, in 2022, I was offered the position to come to Wits. But before then, I learned how to do my own field trips, some excursions. I began new collaborations overseas, and I started developing my career at the museum. And I even dragged my family in to come to field excursions and that. This is an old photograph of my daughter. But I do want to mention somebody who worked with me at the National Museum. His name is John Naipuli. And he was a fossil preparator there. He was already over the age of 65 when I joined the museum, and he stayed for nearly 15 years with me at the museum, teaching me everything they need to know about fieldwork and fossil preparation. He was probably the best fossil finder and fossil preparator in the country, even winning an international award in 2011 at our largest paleontology conference, the Society of Vertebrate Paleontology, for all of his efforts. Sadly, he passed away during COVID, but I do want to make a special mention that I really appreciated his support. He used to say, if I'd freak out in the field and something wasn't going according to plan or something had broken down, he'd say to me, "Bikkie, bikkie, Mark Bayer." And I know there are people online who don't speak Afrikaans. It just means bit by bit we'll get there. He was always telling me, calm down, we'll get to where you need to be, and we always did. So I joined Genus and the ESI in beginning of 2023, and I began putting together my bone histology laboratory, and I got inundated with postgraduate students, which I'm very happy about because the museum did not offer many opportunities to engage with students. And here I'm very happy to have a whole swath of them. These are some of my PhD students. I have to mention Kimmy Chappell. She has graduated, so she's not a current student, but she has a position at Stony Brook University in the States, so she has gone on to great heights, and she is somebody to watch because she's going to be a world leader one day. So she is really my pride and joy. But that's a brief history of myself. What do I actually do? And at least now my family can see properly what I do. I probably don't have to convince most of this audience why it is important to study past mass extinctions. We want to find out which species go extinct and why. We want to have a look at the survivors, what advantages they may have had, pre-adaptations, what made them so special that they managed to survive when others did not. We want to look at the new lineages that radiated. So some of these species would have become extremely abundant. So why? What was so special about them? And we want to know how long it takes for an ecosystem to recover from a mass extinction. And I'm pretty sure I don't have to convince this audience that we are going through a current mass extinction. There have been 5 major mass extinctions in Earth's history. We are now going through the 6th one. And for this picture, you don't really need to look at the details. I just want to point out the areas in red, where these are the species that are currently threatened. 13% of birds, 25% of mammals, and 41% of amphibians. We are losing species every day, and it is the speed of the extinction that is happening that is quite frightening. A lot of the climate scientists watching this will be very familiar with this, but this is just to give you a background. I think we're all aware that greenhouse gases are affecting our environment. They're made up mostly of carbon dioxide, they're in red, and in yellow, methane, and a couple of other greenhouse gases. And all of these work together to increase the global temperature. So if we look at a brief trend, and I'm saying this is very brief because paleontologists do not count 1850 as old, we can see just during the industrial evolution that there has been a massive change in our global temperature. The green line is fairly stable, and those are natural drivers that affect our global temperature. But the red line is where human effects are involved, and you see it has started to climb very rapidly, and this graph actually needs to be updated because we are actually now at the 1.5 mark in 2025. So the red mark actually comes up here. So I want to show you what you're in for. So I'm going to talk about probably the most catastrophic mass extinction that happened both on land and in water. It's called the Permo-Triassic mass extinction. It occurred about 252 million years ago. And when we look at extinctions, when we study them, what do we actually do? Well, we go out into the field and we look at a sequence that preserves this time period, and we have a look at the sedimentology, so we can recreate the environment, so we can look at changes in climate, we can see if the environment became hotter or colder or wetter or drier. We look at the geochemistry as well, so we can look at isotopes, and that can tell us something about the global temperature as well. And of course, we look for lots of fossils. So we look at the fossil plants and animals, and we see who disappears, who appears in the post-extinction environment, who is abundant, and who is rare. So what we do find with the PT extinction is that there was a massive change in climate. We went from something like Mississippi-sized rivers. So if you can imagine the South African Karoo Basin, which, by the way, has the best, most complete preserved sequence of the PT extinction on land anywhere in the world. So if you want to study the effects on land for this extinction, you come here. So everybody from around the world actually comes here to have a look at our sequences, our rocks, and our fossils. So you have to imagine the Karoo Basin as something actually quite lush with Mississippi-sized rivers, permanent rivers, lush vegetation. It was a very healthy ecosystem. And we went from that to something like this, a much drier landscape, but we also had flash floods. So they were ephemeral rivers. In other words, they dried up. They did not stay permanent throughout the year. So animals had to find water. So they had to battle with drought, but they also had to battle with flash floods. So you're getting these extremes from both sides. So what you're getting is a very unpredictable environment. And the key word here is unpredictable. Not just going in one direction slowly, but being unpredictable. It makes it very difficult for organisms to adapt in such a changing environment. So on the left, we're looking at a late Permian ecosystem where everything was healthy. That little creature there, it's my favorite creature called Lystrosaurus. It's a therapsid, an ancestor of mammals. And I have to put a Lystrosaurus in here somewhere because he's my favorite creature and everybody in my community knows it. So I have to give a shout out to him. But unfortunately, although he survived the extinction, you can see the environment that he was left with. It wasn't a very pleasant environment, and it was a very difficult environment to survive in. So I go to sections such as these. This is one of my favorite sites, and we collect fossils, and we do geochemical analysis, and we look at the sedimentology, and we very carefully find the fossils, ones that disappear, and look for the ones that appear during the post-extinction environment, and we have a look at those changes. So I'm interested in this whole extinction interval. What we do find is that burrows seem to have played a very important role in the PT extinction, in those survivors. What we find is a dramatic increase in burrows. So what we find are fossilized burrows, and we find usually they're about that wide, and they are filled with Lystrosaurus skeletons. So when we're very lucky, we find a terminal chamber, which is where the animal would end at the end of the tunnel, and they would curl up and get preserved there. And we have found many Lystrosaurus specimens in burrows of that size. But it wasn't just Lystrosaurus. We find smaller burrows as well, such as this one over here. These are made by other therapsids, such as cynodonts and therocophalians, and we know that amphibians also burrowed. So it really does seem to be a case that the animals that were doing well were the ones that had burrows. Why would burrows be advantageous? Well, they offer a buffer against extreme temperatures. So if it's very cold or very hot, the burrow acts as a buffer against that. It's also a very safe place to raise your young, to store your food. And if you are a burrower, you are making a very important contribution to the ecosystem because you become an ecosystem engineer and you turn up the soil, you open up roots, food for other creatures, and you even provide safe spaces for them as well. So you become a very important part of the ecosystem. I'm also interested in the biology of these animals that pass through these extinctions. I work on something called osteohistology. It's the bone microstructure of fossil and modern bone. Just to briefly explain, we are very, very lucky in that most instances fossil bone preserves well enough that although the organic components disintegrate, the position of the collagen fibers, the blood vessels, and the bone cells all stay in their position. And by looking at modern animals, so over here this is a modern deer, and this is a dinosaur, we can actually interpret what we are seeing through the bone histology. So we can look at different tissues. We can see how fast or slow the animal was growing. We can see how old it was when it died. We can determine if it was a juvenile or a fully grown adult. We can determine even perhaps when reproductive maturity may have occurred. And I just want to point out, even if you don't know anything about bone histology, you can see a difference between this hippo, which is amphibious, and this badger, which is a burrower. So even the ecology of the animals play an important role on shaping the bone histology and the bone microanatomy. But for the purpose tonight, I just want to point out these little arrows. They are indicating annual growth marks. So what happens when it's a bad season, a dry or cold season during the winter? A growth mark is deposited. Basically, the animal stops growing, and then it continues growing again in the better season. And you can actually count those annual growth marks and find out how old the animal is. When these growth marks become very, very close together, you can see that there is very little bone tissue deposited during each good season, which means that the growth is slowing down and the animal is basically reaching maximum size and it's an adult. So what my colleagues and I did a few years ago is we took osteohistology as a tool and we looked at therapsids passing through the PT extinction. Now for this purpose, all you really need to know is that the blue part is Permian before the extinction. The red part is the early Triassic, the post-extinction environment. And all you really need to do is count these arrows. So these arrows represent annual growth marks, and you can see that there are a lot more of them in the taxa that are from the Permian than the Triassic. So what we actually found was that the Permian taxa within these healthy ecosystems were growing to later stages in life, subadults, adults, etc. And the early Triassic ones were dying very young. In fact, most of the taxa would not even make 1 year. A couple of them would do so occasionally, but they were dying at very young stages, and they were growing rapidly. So if we put this into context, all of the ones in blue here from the Permian, these were the ones that were growing to older ages, and the ones in red here during the early Triassic, these are the ones that were growing very fast, but they were dying very young. And what they had to do was bring their reproductive maturity forward in order to breed. And what we see here in Lystrosaurus, there are a couple of different species of Lystrosaurus, but as a genus it survived, but it became very abundant in the post-extinction system during this extinction. And we wanted to know why. And basically, Lystrosaurus bred like bunnies. So they did not get to very old. There are very, very, very few. In fact, there's about 2 that I know of that would reach subadult status. We have thousands of Lystrosaurus in this country and in collections outside of the country. And from their size and from doing work on the bone histology of this taxon, I know that we have never found a fully grown Triassic Lystrosaurus. So they were bringing their reproductive maturity earlier in life so that they could breed. They were so abundant, but then they were dying after about 2 years old. And we see this in a lot of other therapsids as well, not just in Lystrosaurus. They were dying young. And it's only in the middle Triassic do we start to see these patterns change again. So you can see the blue pattern coming through. So we see the typical Permian pattern where animals are reaching later stages in life. They could delay their reproductive maturity. There's still a bit of a mixture, but we at least start to see signs of a healthier ecosystem. So it was pretty great that we could use osteohistology to assess the growth strategies of the taxa through this extinction. Now, there are still many debates surrounding this extinction, and for brevity, I'm going to concentrate on one today because it's one of our active projects. One of the biggest debates is where to place the Perma-Triassic boundary. Now, the marine extinction has been very well dated. There's a section in China that has been radiometrically dated, and we have very good dates for it. But we don't really have radiometric dates for the terrestrial extinction. So we don't really know where the PT boundary is in South Africa. We know exactly where the extinction interval is, but where exactly the actual dates, the time is in there is still up for debate. And people want to know what the date is because they want to know, did it happen at the same time as the marine extinctions? Did it happen before or after? So we have this funny patch at the moment where orange equals Permian and purple equals Triassic, and there's a big fight at the moment going on as to where we draw the line for the base of the Triassic. Now, what you really want to do, which is what happened with the marine section, is you want to find a pristine ash bed. There are crystals in the ash called zircons, and we can use those to date, get really, really accurate dates for strata. And the marine extinction is dated between 251.8 to 251.9 million years ago. And these are the error bars here. They're actually quite low, geologically speaking. Unfortunately, this time period in South Africa has not revealed any pristine ash beds, and that is your number one prize. So we've had to go for the second best thing, looking at detrital zircons. Now, what these are, we still got zircons, crystals from different volcanic eruptions, but they will be embedded with other zircons from other ages, from older volcanic eruptions. So you're getting almost like a dirty sample. So it's not the perfect sample with the most accurate dates, but at least you're getting a ballpark and you can start looking at a pattern. So my colleagues and I found a sample just below where we currently place the PCB, and we dated the sample to 251.7, which is a Triassic date, but it's about 8 meters below where we placed the boundary, and it's within this ballpark. So at least we were somewhere in that area for the marine extinction. Currently, we place the Permo-Triassic boundary based on geology, on the changes in sedimentology, and the changes in the animals and plants. 2020 was quite a busy year, though, because just a few months after we published our date, Bob Gastaldo and colleagues published another date. They looked at a sample just above the boundary, and they found a Permian date. So you can see these two sections are very different, where we consider the PCB to be over here, and Bob Gastaldo and colleagues consider it to be higher up. This is all the extinction intervals, so it doesn't really matter where the date is, but we want to try and see what is going on in terms of the date of the marine section. So this was taken from one of my sites, and I knew exactly where Bob had taken the section, and I can understand why I even thought it was a pristine ash bed. We all got excited about it, but unfortunately it's not. We redated the sample, and we got a Triassic date of 251.65. So we can already see that because we are getting different dates from this little area here, that this is not a pristine ash bed. What we need to do is we need to do a type of analysis called LA-ISO-PMS. I won't go into the details, but you're basically looking for a curve with a huge spike. And the cleaner the spike, where you do not get anything else, all of these other dates, the better. And that can show you whether you have a pristine ash bed or not. If you have other dates like this one, and even though this one looks pretty clean, there are other dates in here, it is not an ash bed. So these are all detritals. So until we find a pristine ash bed, and we will continue looking, we've got to try and find another method for figuring out where we are in South Africa. And this is what my colleagues and I are doing at the moment. We are taking samples through sequences, through many, many sites in the Karoo Basin, from the Permian to the Triassic, and we are using the detrital zircons to get a pattern. So what you can do is you can actually model and have a look at the dates and look at the trend. And I've made those dates really, really small because it's not published yet. And my collaborator might kill me, so they are very small, but basically this is the type of thing that we are doing. And we will be adding more and more samples, and at least I can say that our preliminary results suggest that we are correct where we are currently placing the PCB in South Africa. I'm moving on to another mass extinction, the end-Triassic mass extinction. This extinction is interesting because it also was caused by volcanic eruptions. So everybody pretty much agrees that the PT extinction was caused by volcanic eruptions that occurred in an area that is now Russia. And the ETE was also caused by volcanic eruptions. So I wanted to know, were these animals acting in a similar manner? Did they show the similar patterns for survival? Who was going extinct? I wanted to know what the patterns were and what the differences were. And I'm going to say again that luckily enough, South Africa yet again comes back on top as having the best fossil record of this time period anywhere on the planet. There are other sequences that contain the ETE, and they have been well studied with sedimentology, geochemistry, etc., but there are very, very few body fossils. So people are mainly relying on trace fossils, on footprints, to determine what species were living around that time. But we don't have to do that. What we have is a formation called the Elliott Formation, the lower Elliott and the upper Elliott. And I just want to show you here that you can see there are different animals in each region. And the ETE is running somewhere along here. We also don't have pristine ash beds for this time period, but scientists are looking at detritus to try and get a trend as well to see where we are in the dating. So what do we see? Yet again, we see changes in the climate, again from the lush environment with the permanent large rivers to this very unpredictable climate. It became very hot, it became dry, ephemeral rivers, braided streams. It was a difficult environment in which to live. So I started studying a very interesting group of animals called the Sauropodomorpha. These are the ancestors of those very large sauropods with the long necks and the long tails that you've seen in Jurassic Park movies, etc. Now, these are the smaller bodied ancestors, so they were smaller in size and they were also bipedal. It's only during their evolution that they became quadrupedal. So they went from standing on 2 legs to standing on 4 legs. And all the larger sauropods are quadrupedal as well. So I wanted to have a look at their early growth because we have some of the earliest sauropodomorphs in South Africa. And what my old student, Kimmy Chappelle, did during her PhD is she took on the enormous task of looking at a very special dinosaur. It's an iconic dinosaur for South Africa called Massospondylus. It's a sauropodomorph that lived during the early Jurassic. So this is during that post-extinction environment, and it is extremely abundant. So she looked at the bone histology of a very large sample size. Because it's so abundant, we could do that. And she basically calculated the amount of bone tissue deposition between each of these annual growth marks. And she did this for different individuals, different elements. And although you can see there is a general curve here in red, you can see that below this, and this is why I wanted to use this in particular, it's just a mess. Because that's really the take-home message here. Usually when we look at an animal, we will find that it'll produce the most bone tissue when it's younger, and it'll produce less and less as it gets older, and then it'll stop growing. And yes, we find that in many individuals of Massospondylus. But we also find something else. So for example, we might find in this quite older individual here, large amounts of bone deposition, then smaller amounts because it's getting older, and then suddenly a massive growth spurt when it's older. And we see this in a lot of individuals. In fact, there is so much individual variation in this dinosaur that we call it extreme developmental plasticity. So what Kimmy had found was this intense variation between individuals. When we look at animals today and we look at different individuals, there is very, very little inter-individual variability, but not in Massospondylus. And it's possible that its ability to adapt, to change, to all sorts of different contrasting environments, to changes in climate, allowed it to actually survive and become very abundant during the early Jurassic. So this is an example of something that actually it survived in that environment and it did very, very well. And it was perhaps because it was able to adapt quickly. So here are the big sauropods. They grew up to 70 tons in weight. And I just want to show you this little graph here that Appledetti and colleagues put out a couple of years ago. It'll help you with the next couple of slides. Basal sauropodomorphs, such as Massospondylus, grow more slowly and they are smaller in size and they have interrupted growth. So they lay down those annual growth marks every year. Our big sauropods, though, grow really, really fast. They grow to massive body sizes. And what's interesting is that they only appear to lay down growth marks later in ontogeny. So for the first couple of years, we actually won't see any growth marks. We'll only see them later. So there is a period of continued growth, which is unusual. So Appledetti and colleagues looked at Lysimsaurids. These are the sister taxa to the true sauropods. So over here we have the light blue, which are the basal sauropodomorphs, and in the dark purple we have the sauropod dinosaurs. And Appledetti and colleagues found that these dinosaurs, they were growing to similar sizes of sauropods, and they were growing very fast. So they were already growing as rapidly as sauropods, but they still had interrupted growth. So my colleagues and I decided to look at something further down on the tree for some South African dinosaurs. And what we found is, so here are the lisnosaurids in this aquamarine, growing as fast as sauropod dinosaurs, but still with interrupted growth. Ardonyx and Cephaloponosaurus were much smaller, and when I mean much smaller, I'm still talking about 1 to 2 tons, but for dinosaurs, for sauropods at least, that's really small. So they were also growing really rapidly, but it was not in order to reach large body sizes. So what we found is that that special rapid growth that sauropods had was already present in the more basal sauropodomorphs in the smaller taxa. So it was selected for another reason before these animals got really large. And the rapid growth was co-opted by the large sauropods to be able to grow quickly to reach those massive body sizes. I'm just going to give a bit of detail here because a friend of mine from the States is watching tonight, Christina Curry-Rogers. She's one of the top paleohistologists in the world, and she works on sauropod osteohistology, so she'll be excited to see this. We are looking at so many different new species that are coming out of the Elliott Formation, mostly due to Jonas Schwanier and his colleagues and the fieldwork they've been doing. They have been finding so many new species of dinosaur from the late Triassic. So what we are looking at the moment is this is a phylogenetic tree of all the sauropodomorphs that are known with bone histology data. This is not all of the sauropodomorphs, just the ones where we have bone microstructure data. And actually, it's quite a lot. We now have 38 species, which in paleontology terms is actually a very large sample size. And we have them ranging from the late Triassic through to the early Jurassic. And what's really great is that those in red come from Southern Africa. So Musungu over here comes from Zimbabwe, Kadumadumo comes from Lesotho, everything else in red comes from South Africa. So you can actually see how important South Africa's role is in looking at the early growth of sauropod dinosaurs. So at the moment we have 152 limb bones, and I think only Christy's going to understand how much work that is because every single element has to be very carefully analyzed. And just to show you the kind of stuff that we are doing, I just took a range of different tibiae. So these are 4 different species, and what we do is we trace the annual growth marks, and we take that circumference and we plot it on a curve. So just to make it easier for you, this is the original graph that I showed you. What are we finding with those dinosaurs that where we've traced the growth marks? Here is Musungu, which is a more basal sauropodomorph, and it's a smaller dinosaur and growing slower. Here is Cephaloponosaurus, which is the red here, which is what we expect, growing more quickly than Musungu and reaching slightly bigger body sizes. We have a new species of lismosaurid in South Africa, in that aquamarine, growing larger and faster. And now we have a completely new species that we don't really know where it fits in. It's a sauropodiform, so it's more basal than these lismosaurids, but it's growing just as fast as the lismosaurids. And what we need to do now is that what Jonah is pulling out is more and more sauropodomorphs that are actually quite large, larger than I expected. And it may be that these larger dinosaurs are growing quicker than expected because of larger body sizes. So what we are doing now is teasing out the effect of body size, because we want to look at the evolution of the change in body size in sauropodomorphs, but we have to be careful about the effect of body size. So it might be that the phylogenetic signal is completely taken away when we look at body size, and body size becomes a more important factor in allowing in looking at these animals' growth than the phylogeny. But that is a project that we are busy working on at the moment. So I've told you something about the dinosaurs passing through the ETE. Now I want to move to the crocodilians. So the ancestors of crocodilians are called crocodilomorphs, and in a couple of years ago, in Jonah's fieldwork, they pulled out a really big crocodilomorph. He's a giant reptile, and he is a little bit odd to us because we're used to crocodilians being quadrupedal. This animal was bipedal, so it was walking on 2 legs, but it was a couple of meters in length, and it was gigantic compared to some of its descendants. So we looked at the bone histology of this animal. It lived in the late Triassic, and I'm not going to give you the boring details of all the technical details of the histology. I just want to point out, because this animal contains a combination of something that's quite special. So it contains a bone tissue, we call it parallel fibered bone, which is somewhere in between fast and slow growing. And here I can see with these green lines that it was 3 years old, but that the amount of bone deposition over here is less than over here. So growth was slowing down. So it was becoming a sub-adult. It wasn't a juvenile anymore, but it hadn't actually reached its maximum size, so this animal was going to get even larger. But what is interesting here is the orientation of these big holes over here. These are the spaces for the blood vessels. And usually when we see them in a circumferential pattern like this, we see this in very rapidly growing taxa. So what we're getting is the rapid vascular orientation mixed with the slower growing bone tissue. So if we put this into context, here we have, these are all reptiles called archosaurs. Here are our dinosaurs growing very, very quickly. Red means fast growing, black means slow growing, and somewhere in between is purple. The early Jurassic crocodiliforms over here all grew slowly, and they were all very, very small. And we know today, looking at crocodilians, that they grow slowly, they have low metabolic rates, but we know they came from faster growing ancestors because we know they came from this group of archosaurs. But we didn't know where the actual transition from fast to slow growth happened. And in finding this animal here in the late Triassic, it's before the ETE, we could see that growth rates were beginning to decline even in the larger crocodilomorphs. So during this stage, these animals became smaller and smaller, they grew slower and slower, and they survived the ETE. So what do we have here? We have the dinosaurs growing really fast and really big. They survived the ETE. We have the crocodilomorphs becoming smaller and smaller and growing slower and slower, and they survived the ETE. So what do we have here is my mass extinction program where I'm comparing the Perma-Triassic mass extinction to the end Triassic extinction and basically everything in between. I'm looking at the Triassic. There is an event called the Carnian-Pluville event that occurred in the Triassic. I won't go into it now. It was a smaller event, and people debate as to whether it was global, but I would like to include that in my analyses in future. All you really need to see here is that the black part here, these are all therapsids, so they are the ancestors of mammals. They became quite large in the Triassic, and then they became really, really, really small, like the size of mice, to the very first true mammals in the earliest Jurassic. This white section here are all of the archosaur reptiles. And you can see that the dinosaurs and the crocodilomorphs survived the extinction quite well. There were a lot of other reptiles that did not. They went extinct. And what's very cool here is that we have small, slow-growing taxa passing the extinction. We have large, rapidly growing taxa passing the extinction. And although I cannot say what's happened here, about a month ago we went to the synchrotron, to the ESRF in Grenoble, because any limb bones from these really, really tiny mammaliaforms cannot be cut up. So we scanned them using the synchrotron, and we've gotten some great bone histology structure, and it's given us a very unexpected, exciting result. So unfortunately, I can't say anything now because my collaborators will kill me, but watch the space. This is going to be a very exciting and really cool paper, so I'm very excited about that. But now that's looking at research in the past. So what does that mean for us now? Why should we care looking back 250 million years ago? Because we are sitting somewhere over here. So you can see, this is just going back to the end Cretaceous extinction, that global temperatures have fluctuated and been higher than they are right now. And we have graphs of the global temperatures going back to 500 million years or more. But what we want to do is we want to predict what is going to happen. So let me zoom in on that now. So climate scientists look at this, they look at the past, and they try and predict the trends of what we might see into the future. And they say by 2100, the climate will be something like the Miocene climate optimum, which is something we had about 16 million years ago. So it will be hotter, but what we will have is the North and South poles will no longer be covered by ice. What happens when that happens? Well, it changes the ocean circulation. You have more ice melting into the water, it changes the salinity. It will cause many, many marine organisms to go extinct. The ocean circulation is also tied very closely to our whole planet's climate. So it will change the climate, which will then affect our terrestrial ecosystems as well. So we can look forward to some extinctions there. They say that if conditions are not stabilized by 2250, by 2300, we will reach the early Eocene hothouse of the world. Now, it was much hotter back then, and there were selected extinctions, but it's not a mass extinction. But the difference is that it's not just about getting hotter over a period of time or drier. If it's over a long enough period of time, animals and plants and organisms can adapt. It's the speed at which it occurs. So the scariness is the unpredictability, not allowing animals to adapt. If you give them enough time, perhaps they can adapt to changing conditions. So what are we likely to see? Well, looking back from the fossil record, we know that certain taxa are likely to survive. We call them pest and weed ecology. So at the PT extinction, it was Lystrosaurus. At the ETE, it was the Massospondylus dinosaur. So what is likely to survive and do really, really well in human-dominated environments? Rats. So basically, rodents are probably going to do really, really well. And of course, they breed like bunnies. So we are probably going to see a lot more of rodents and similar creatures. We are also going to see a decrease in biodiversity. And why is this a problem? Because I know I'm also speaking to the next generation. And kids always say to me, why do we care about biodiversity? Why do we care about the past? If something comes and wipes out these flowers here and there's nothing else, what are you going to eat? So you have to have genetic variation. You have to have biodiversity. This is why cloning doesn't work, because there's not enough genetic variation. So if you are left with just a few species and something comes and wipes the main dominant species out, what are you going to be left with? So unfortunately, it is very sad that, and I'm sure everybody already knows this, we are going to look at the disappearance of the large predators and the large herbivores. And we see this in the PT extinction as well. We had incredible predators. They were like saber-toothed cats called Gorgonopsians and huge herbivores. So they would have been similar to the cows that we are and zebras, etc., that we're used to today. And unfortunately, our elephants and our rhinos are going extinct, and so are the large cats, our large predators. And we're also doing something really stupid, which is attacking our powerhouses. So in areas such as the Amazon rainforest, that is literally the lung of Earth. And we are busy destroying habitats such as these. So these plants are creating oxygen for us, and we are destroying these habitats. We are destroying these powerhouses. And this is going to have serious consequences for us. So what might we see? Well, we might see, so we're starting off with a lot of biodiversity, big and small taxa, all sorts of different shapes and sizes. We start to see an extinction of a lot of these taxa, especially the larger ones. We're left with just a few species that might make it. This species will go on and radiate and make more species. So eventually you will get a whole different ecosystem structure. We may or may not be a part of that. But we will see changes, but to get through this passage of suffering is you don't really want to go through a PT extinction, trust me. So what are we looking at? Right now we are looking at a very unstable planet. There are climate extremes. I'm sure everybody has noticed that the natural disasters are getting worse. The flooding is much higher than in older records. The droughts are worse. The typhoons are worse. The hurricanes are stronger. So we don't just get something with an increase in global temperature. We don't just get hotter and drier, for example. We will get more flooding on one side of the planet, and we'll get more droughts on the other. And what we're looking at is a seesaw. So we are looking at increased unpredictability, and it is happening like that. It is the speed at which this is happening that we've never seen in any other past extinction. So we will see a decrease in biotic diversity, and as I say, that is going to mean the loss of our food, of our crops, of our animals. Economists suggest that there will be a decrease in global GDP by 18%, which means the costs of living are going to go up. It's going to take much, much more to keep your standard of living. We are going to see more and more people poorer and starving. There are 8 billion people in the world. There are too many people, and there are a lot to feed. But I didn't want to end on a really bad note. We're all going to die, and this is the end of the world, especially as I have my daughter sitting in the audience. So what can they look forward to? And this is a shout out to the new generation, the generation of students right now, and the school kids as well, is that it's not too late. So they estimated that with the PT extinction, there was an increase of about 8 to 10 degrees Celsius, and we're sitting at 1.5. There is still time to do something. Yes, we didn't make our 1 degree mark, and we are heading towards 2 degrees, but if we all work together, we cannot prevent the extinction that is happening now, but we can slow it down. Slowing it down gives organisms a chance to adapt to these changing conditions and may even help us and our descendants to suffer less. Because in 200 or 300 years, it's our great-great-great-great-grandchildren, or so, a couple of generations, who are going to be looking at us and saying that we were responsible for this because we started it with the Industrial Revolution, and we were not doing enough to prevent it. And we can prevent it. We can decrease our greenhouse emissions. We can decrease habitat loss, but we all have to work together in systems such as the Paris Agreement. I find it remarkable where countries so different can come together for a single cause and fight together to save the planet. Leaving agreements such as this is insane because no country lives in a bubble. What happens on one side of the planet is going to happen everywhere. Each country does not live in a vacuum. We all live on one planet. And if the planet goes, we all go. So it is vital that those coming up in the next generations realize how important it is to do something now, because we are at the tipping point now, and we can really do something to prevent what happened 250 million years ago. Thank you.
Thank you very much indeed, Professor Botha, especially for your very impassioned plea at the end. Yes, I agree with you. We have too many people on this planet, and that, of course, is a worry and putting enormous pressures on our ecosystems. From dolphins to paleontology, that was really a lovely journey that you shared with us. So thank you for that, and totally agree. We've got to understand the past to plan for the future. Interestingly, just a week ago, I was out at the Golden Gate National Park with Professor Joanna Chouanier and Dr. Kimmy Chappell, who I understand now studied with you. So I did get to see Massospondylus looming very large out at the Dinosaur Center, so I see the connection, but that is really a wonderful place to be. Colleagues, if you have the chance to get over to that part of the world, and of course our colleagues have been quite involved in setting this up. Thank you very much for explaining some pretty difficult concepts in ways that are very understandable. Especially the ideas around osteohistology in understanding the rates of growth of these organisms. I certainly understood that fairly well. Although these terms are just almost impossible for me to say. I've got to say that the hardest thing for me is to read out some of the names, the thesis titles at graduation. Whenever there's a thesis out of the ESI, I do shudder with a bit of uncertainty. So thank you, and thank you also for recognizing your preparatory, the late John Pulley's what I wrote down here. So I've come to appreciate the extremely important role that our preparators play in the work that you do. So thank you very much for remembering him so well. So as a token of appreciation, I do have from the Deputy Vice Chancellor, Professor Ian John Rowell, we do have a token of appreciation to share with you. Thank you now. Yes, giving an inaugural address is certainly something very special. So colleagues, it's now my pleasure to call on Professor Joanna Chouanier, who will move the vote of thanks in a moment. He will thereafter call upon Professor Marion Bamford, who will close the proceedings thereafter. Now, Professor Joanna Chouanier is a renowned dinosaur paleontologist. You've already heard his name several times. During the talk, he got his PhD from the George Washington University in the United States. focusing on theropod dinosaurs in China before moving to WITS in 2012. He conducted a postdoctoral research at the American Museum of Natural History and led expeditions across Mongolia, China, South Africa, and the USA. Professor Chouinier's work concentrates on the systematics, taxonomy, and paleobiology of dinosaurs and basal archosaurs, I hope I say that right, especially base sauropodomorphs and the Triassic-Jurassic faunal transition. He has co-described numerous species using advanced techniques such as CT and synchrotron-based scanning. Professor Chouinier continues to do extensive field work in the Karoo Basin and Zimbabwe, seeking to unravel early dinosaur evolution. Professor Jonas Chouinier, please move the vote of thanks.
Thanks, Prof. Chetty. I'm going to depart from my prepared remarks, as I so often do, to begin by thanking Lystrosaurus and Massospondylus for their profound sacrifice that allows us to understand these extinctions in deep time. And I'm going to ask you guys actually to hold your applause until the end, just in the interest of time. So thank you, Lystrosaurus and Massospondylus. I've known Prof. Botha for many years, and we've actually had a lot of fun together doing field trips and going on trips to the European Synchrotron Radiation Facility. And she very awkwardly asked me to thank myself, standing here, and I've decided that I'm going to do that by thanking myself for not showing many embarrassing pictures I have of you on the screen. But if you would like them, I'll make them available for a very low price after this ends. Seriously, though, let's see. And lastly, on that topic, I wanted to thank you, Professor Botha, for sharing so many slides containing data I've been asking you for months. It was a pleasure to see some of these things on the big screen, and looking forward to writing those papers with you. But seriously, I'm actually delighted to see so many of the professors, current and former students, in the audience tonight, ranging from undergraduate students to postdoctoral fellows. I just want to remind you that you're all her pride and joy, not just Kimmy. And thank you for attending this lecture. It's really, you students really enrich our academic lives so much, and it's been such a pleasure sharing the academic journey with you. And thank you for your curiosity and your unique perspectives, which we hope continue for a long time. I'd like to thank Professor Botha's family, Kayla, Daryl, and Drew, who are in attendance tonight, for their unwavering support during her many years of education and research. Your patience, love, and understanding keep her grounded. And you play a larger role in her career maybe than you realize. And I think that was evidenced by the cool old photos you showed us. You've been there from the start and will continue to be there. So thank you very much. I'd like to thank the academic peers and role models who've helped shape Professor Botha's career, particularly her former manager at the National Museum, Dr. Rick Nuttall, who I've been out with actually in the field myself at Golden Gate. He's a wonderful guy, but many of you are in attendance tonight, and your guidance has been invaluable for her scientific journey. I'd also like to thank the stewards of WITS's academic community who are here tonight, including Head of School Professor Marion Bamford, Dean of Science Professor Nithya Chetty, and the DVC of Systems and Operations Professor Ian Jandrell, who gets a special mention for pronouncing my name correctly on his first try. Guys, it's actually not very easy steering a bunch of academics, and we realize that, but we appreciate your hard work in keeping the whole show going, so thank you very much. I would finally like to thank everyone in the audience this evening for sharing your time and attention on this joyous occasion of welcoming Professor Botha as the new professor of paleontology at WITS University. And now you may issue your applause. Without further ado, I'd like to welcome Professor Marion Bamford to the stage to issue the closing remarks. Professor Bamford is the head of school of the Evolutionary Studies Institute.
Right, so Jonah has thanked all of the relevant people, and that's a large number. Of course, not all the names have been given. But I would like to thank WITS. And WITS has supported the ESI, formerly the Bernard Price Institute, for almost 75 years. This year is our 75th anniversary. So we have a long legacy of researchers, students. I myself am a former student, and the students here might be future members of the ESI staff as time goes on. But we have a long legacy of research, of our passion for the science. I'm sure that has come through very clearly with Professor Botha's well-articulated talk. So thank you to everybody for all of that. And let's keep it going. We've only done it for 75 years. How many more millions of years must we work for? Thank you. Would the people stand while the academic procession leaves, and then follow us out into the foyer to join us for some snacks where you can personally talk to Professor Botha. Thank you.